So happy new year to all of you. And today's class will be estimation of blood glucose by glucose oxidase peroxidase method. Okay, it is one of the most important biochemical point of view test. Okay, like till now we have done what? Glucose estimation from urine, and we have learned about diabetes and all other diseases. Okay, where you can find the blood glucose level. Blood glucose, till now we have done urine glucose level, right? And diabetes and related topics. Clinically, why are they important? What are the importance? Okay, so this 1 week we will be teaching you. First is that glucose estimation. Another one will be protein estimation. Okay, and mind my words, both these tests will be there in your upcoming formative assessment as well as in your internal assessment. Okay, and this holds chunk of mark. Okay, each of the test. So you have to perform the test perfectly. Theoretically this is very easy, nothing much to say. It depends upon one law. Okay, that has already been covered with you, that is the Beers and Lambert's law, right? That is by colorimetry. So this instrument you have already worked on, so you know this instrument, this is the colorimeter, and by using the same principle, the same Beers and Lambert's law. Everybody remembers Beers and Lambert's law? Somebody tell me what is the law? Absorption is? Beers law is? B directly proportional to C. Yeah, C is concentration, and I or L is length. Okay, that is also directly proportional. Over here the length is fixed, so that is why your absorption is directly proportional to the concentration. More the concentration, more will be the absorption. Okay, so that is how this this test also depends on the same formula, the same principle. Okay, okay. Basically what we do is we take 3 test tubes. Okay, right now I am just giving you a rough idea, then after that I will go into the topic proper. We take 3 test tubes. In one test tube we will take a blank sample. Blank sample means we will take only distilled water. Okay, another one will be standard. Standard means we will take one sample, the concentration in that sample we will know, and we will take another test tube, that is a test. Okay, in that sample we don't know the concentration of the blood sugar, and by doing this colorimeter we will estimate the blood glucose level. Okay, let's see. So introduction is, first of all you have to do the sample collection. Okay, so if we are taking sample collection, anybody has any idea what is the most common place to take blood? This is which artery? Artery or vein? Vein. Okay. Okay, median cubital vein. Okay, why we are not using artery?
Deep, so deep artery.
Yeah, one is artery is deep. Okay, another reason is there why we are not using artery. In case of glucose, at least we are not using arterial blood. We try to use the yeah, but we can count with that. If the flow is high also, we can use narrow needles that can take the blood. Okay, we do test of arterial blood as well. This is not that we don't do the arterial blood test. We do ABG, arterial blood gas test, where the pH and other bicarbs and what is the oxygen saturation is how much in the blood, that we can check from the arterial blood. So we can do the arterial blood test, but in case of blood sugar we don't take the arterial blood, because arterial blood can give you a false result. Why? Okay, in the arterial blood what happens, the blood glucose level can be higher. Why? Because the arterial blood is taking the nutrition to the peripheral tissues. Over there the glucose is getting utilized, and then the arterial blood is coming back as a venous blood, and that is what we are measuring, that how much amount is circulating in the blood, right? We are not estimating how much glucose was there from the systemic circulation that came into the artery, because the artery will take the blood to the periphery. Over there the peripheral tissues will take up some bit of the glucose to metabolize and get nutrition, and rest of the glucose will keep on circulating in the vein. So that is why whenever we are doing the arterial blood test, we can get around 35 milligram per deciliter of extra blood glucose level. Okay, but in the venous we will get the appropriate one, that is the circulating blood glucose level. That is what we are measuring, how much amount of glucose is circulating. Prior use, if you are measuring, then you will get a higher concentration. Got it? Anybody has any doubt in that? So that is why we are not taking the arterial blood, we are taking the blood from the veins only. Okay, now which portion of the blood we are using? As soon as you take the blood sample from the patient, there are 2 components. One is the blood, so there are 2 constituents. One is the blood cell and the plasma. Okay, that is also known as the serum. Okay, so basically we take the blood and then we centrifuge it. Okay, the constituents of the blood like RBC, WBC, everything settles down, and the serum is floating above, and that serum we take it. okay, and then we measure the blood glucose level, and that is the serum blood glucose level. That is why we say as the serum blood glucose level. Okay, that's all. So plasma is preferred sample for estimation of blood glucose, and whenever you are taking a blood sample, we cannot just take in any container. There are some strict guidelines for each and every blood sample to take. For blood glucose, we take gray container, means the container which is capped with a gray color cap. So all the containers are already color coded. That you will get to know in detail when you will go into the pathology. Okay, so there are many more colors: green, orange, red, yellow, everything is there. But for the blood glucose level, we are using the gray vacuoteners. Okay, the vacuoteners means there is already a negative pressure which is created into the tube where you are collecting the blood. Okay, and the cap is of gray color. Why it is gray color? Because there are some fixed molecules which is already there inside the container. One is that will avoid the blood to coagulate. That is known as the anticoagulant. Because if you take a blood sample in a normal container, after some time the blood will coagulate. That is the natural property. But if there is anticoagulant, then the blood will not coagulate. Okay, and the blood will remain like that. Because it is not mandatory that you will take the blood right now and immediately you are doing the test. You will take the blood, you will preserve it for some time, and after that probably you are sending it to far away from this place wherever you are taking the blood. So that is why we have to preserve it so that it don't get clotted. So just to avoid that, we add some anticoagulant into it. Okay, and there are some product as well which will avoid glycolysis. There is one step, if you have seen in your theory part, that whenever glucose is getting metabolized, there is one step with the enzyme enolase. Okay, that is happening in the glycolysis pathway. So to avoid that enolase pathway, if you skip the blood like that, the glucose in the blood that is present, that will some bit of it will get metabolized. Okay, and glycolysis will happen, and eventually you will get, let's say the patient's actual blood sugar level is 100, but you will get 80 because some bit of it got glycolized. Okay, to avoid that enolase step, we are using some enzyme that is known as the sodium fluoride. Okay, so vacuoteners, the gray color vacuoteners contains what? One is sodium fluoride to inhibit this step, to inhibit this glycolysis pathway that is done by the enzyme enolase. Okay, so that is what we are doing, one step. And another thing is potassium oxalate. Potassium oxalate is the anticoagulant. Okay, what it is doing? Preserving the blood sample as it is in the liquid form so that it don't coagulate. Okay, everybody understood? So these are the two things that can be asked in your viva as well as during your exam, like which container we are using. That is the gray color container to collect the blood sugar level. To measure the blood sugar level, we collect the blood sample in the gray color container. And what are the constituents in the gray color container? One is the sodium fluoride, another is the potassium oxalate. Potassium oxalate is doing what? Anticoagulant, and sodium fluoride is for the prevent glycolysis. Okay, that's all. Now blood should be collected from the veins, because in fed condition means when I am having food, after having food, like let's say if early in the morning. Okay, if early in the morning you are taking a sample from the artery or from the vein, both will have more or less similar value only. Okay, but as soon as I take food, I am now in a fed condition. Now from my intestine, all the nutrients are getting absorbed. This will come into the blood circulation, and this will be there in the arterial circulation. Why? Because this will go to the tissues now to give the nutrition to the tissues. Now if you take from the artery, you will get a higher value. Okay, but if you take from the veins, because the glucose has been dispersed in the peripheral tissue, now the blood is coming back through the veins. So now if you take from the veins, then you will get a normal circulating value of the glucose. That is why we have to avoid taking arterial blood sample in a fed condition. In the early in the morning, when the patient is having NPO, means nil per oral, for around 8 to 10 hours, that time you will get more or less same only. Okay. Now there are some methods to estimate the blood glucose level. Okay, so what are the methods? One is the enzymatic method. So the names are like this. Enzymatic method means one is glucose oxidase method, another one is the hexokinase method. So this is how the rows are. Okay, now reduction methods are ferricyanide method, ferricyanide-Hoffman's method, copper sulfate method, and the aromatic amines method. Today and in your entire class, you will be requiring to know only the glucose oxidase method. Okay, and rest of the names can be asked in viva, like what are the reduction methods, what are the chemical tests, what are the other tests that we can do. Okay, so these are the things that you have to just know the names. We will be performing only the glucose oxidase test. Okay, why we will not perform the other test, that also I will let you know. Okay, but otherwise this test you have to just know the names of the test. Somebody can ask you, other than this GOD, glucose oxidase peroxidase test, what other test can be done to estimate the blood glucose level? So if they are asking like that, some of you can give an example of hexokinase test or modified King Asatho test or else orthotolodine test. Like that you can say 2, 3, 4 names like that. Okay, somebody is being very specific and asking you what are the chemical tests that can you do, then you have to say only these 2 names like that. Okay, so charts are given over here, you can just write down the names and you have to just by heart the names. Okay, okay. Now glucose oxidase method, this is the test that we will be performing. Okay, first of all, reagents. What are the reagents we are using? Reagents use, what are the reagent? First of all, one is the enzyme reagent, that is we also known as the GOD-POD enzyme reagent. Okay, what are the contents of the enzyme reagent? That is the glucose oxidase, peroxidase, 4-amino pyridine di and phenol in the phosphate buffer. These 4 together is known as the enzyme reagent. Okay, this will be found in one container that will be given to you. These are already pre-made samples. You have to take the samples from here. Okay, that's all. Second is the glucose standard. Glucose standard means I told you about one. We will take one test tube where we will know the concentration of the glucose that is there. So we know one glucose that the standard value is the 100 milligram per deciliter. So this value we know. Okay, and third is the test sample which the glucose level is unknown, that we will be estimating. Okay, and another one is distilled water. I did not mention distilled water, but that is a blank test tube that we will add. Now what we will do is we will take 3 test tubes. Okay, let's say these are 3 test tubes. Okay, now I have marked this one as B means blank, S means standard and T means test tube. Okay, so these 3 test tube I have taken. Now among this I will add this enzyme reagent which is written over there, that is a GOD-POD reagent. Okay, I will take 1 ml of each in each of the test tube. 1 ml of enzyme reagent, 1 ml of enzyme reagent and 1 ml of enzyme reagent in 3 of the test tubes. Okay, then in the blank I will add what? I will add distilled water. It is blank, so it will be distilled water. How much? 10 microliter. This 1 ml is also equal to 1000 microliter. Okay, if in case anybody has any doubt. So this is 1 ml of GOD-POD reagent with 10 microliter of distilled water. Over here 1 ml of GOD-POD reagent along with 10 microliter of glucose standard. Okay, over here 1 ml of GOD-POD reagent with 10 microliters of test samples. Done. Now we will mix this thing, all of this thing. Now thing is, after mixture you have to incubate it. Incubate means you have to give some heat into it. Okay, we don't directly heat it like the way we used to do in case of Benedict's test, right? We will not directly heat it, we will put it inside the incubator or inside the hot air oven. Okay, anywhere you put it and you keep it for 10 minutes. After 10 minutes what will happen? Some reaction will happen. I'll come into the reaction. Some reaction will happen, and if the sample is containing glucose, then there will be some color change. Okay, that will be pink color. By seeing that you can identify with your own eyes that probably the sample is containing glucose, and that is why the color change is there. So with your own eyes you can estimate that whether the glucose is present or not. If there is no color change, means there is no glucose, but eventually we will do the test to estimate the value. Okay, so now these are quantitative tests. Okay, we are here measuring the exact amount of the blood glucose, not like an approximation. In case of Benedict's we were saying like probably 1.5 to 2%, more than 2%, like that. Some range was there. Over here we will not get any range. We will get the exact value of the blood glucose that is present. Okay, so after this mixture we will incubate it for 10 minutes at around 37 degrees centigrade. Once the incubation is done, you take out the sample and then you put in the colorimeter. Now in this colorimeter over here, you know how to do the zero. Okay, how to do the zero? You put a blank sample into it and then make the zero. Your zeroing is done. Once you make the zero, then after that you have to know the lambda max. In case of this test, the lambda max is around 520 nanometer. 520 nanometer. You can perform it as well. No issues, but this is already given. The sample that we are already using in the manufacture, in the use that what do you say? How to use it? That in the introduction or induction. Yeah. So over there, this is already written over there as well. Like in which wavelength you will get the maximum absorbance. Okay, so that is your lambda max. You got already lambda max. So what we will do in the colorimeter, we will fix the wavelength to 520 nanometer, or also known as a green filter. We have to use the green filter. So in this nanometer, now first of all I will put this blank sample. Okay, once I put the blank sample, I will get some absorbance, that is known as the ODB. Let's say I got the ODB as 0.21. Let's say, just an example. Then I will do for the standard as well. Let's say over here there is no glucose, right? Over here glucose is there, so concentration is high. So if the concentration is high, the what will be the absorbance, higher or lower? How come lower? Higher. It is proportional to concentration, right? If there is no glucose, then it is coming like this. If there is glucose is present, obviously it will be little bit higher. Okay, let's say it is 0.35. Just an example. And in the test tube also you will check the absorption. Now here this can be two possibilities. Okay, possibilities are this is more than the standard, or it could be less than the standard. If it is more than the standard, let's say ODT. If it is more than the standard, let's say in the standard we know that the glucose concentration is 100. In the test, the glucose concentration is 200. Let's say 200. Then what will be ODT? More or less? More. Okay. And if it is let's say 80, then ODT will be less. So let's say over here the concentration is 200 only, and you got something around, let's not make it more. Okay. And you got 0.40. This is what you got. Okay, now from there we have to calculate the glucose level. How to calculate? First of all is ODT minus ODB divided by ODS minus OD blank into the standard concentration. The standard concentration is how much? 100. 100. That's all. So if you just put the value, it's just a class 1 mathematics. ODT is 0.40, ODB is 0.21, ODS is 0.35 and minus 0.21 into 100. Right? Can't help it, maths is there, you have to do it. Okay? Let's say 0.19 and over here it comes 0.14 into 100. Just calculate it up, whatever the value comes, that is your blood sugar level, and that easy is it. Okay, basically your part is when you are doing this test, this part of calculation is very easy. Okay, just put the value and solve the equation, that's all, nothing much. The thing that you have to concentrate and you have to be clear about is when you are taking the samples. Okay, whenever using the micropipette, you all know how to use the micropipette, you have all used it. So whenever you are using the micropipette, first of all you take the 3 samples for your betterment, 3 GOD POD samples in all the 3 test tube. That's a separate pipette tip you are using because the pipette tip will be little larger. For the 10 microliter you will be using the narrow microtips. Okay, so when you are using the narrow microtips, let's say somebody who is taking the standard sample first. Okay, first you are taking the sample standard, then you are using the blank. What will happen? There will be mixture of the standard and black and your values will be altered. Either you can use what? You use 3 microtips. One is specifically for blank, one is specifically for standard, one is specifically for test. So that is the ideal way. Okay, but if you are not doing it, in exam somebody has given you only 1 microtip, let's say. Okay, that time you will first fill up the blank with the microtip because that is distilled water. It cannot get mixed with anything. If my blank is mixing up with S, no issues, but S should not mix up. Okay, because once you are taking up in the microtip, then you are taking the sample, then some bit of his glucose can be stick up in the wall. There is no sample, but some bit can be there because we are dealing with microliters. Okay, it's a very less amount. So that is why first you fill up the blank, then you go to the standard, and then you go to the test. Okay, okay, let's see. Over here, method is first take 3 test tubes in the rack. You mark it like. Let's say in the rack these 3 test tubes are there. In the rack only you write 1B, 1S, 1T, so that there is no confusion like which one was B, which one was T, because you have to put it inside the incubator, and by the time after 10 minutes you are taking out from the incubator, you might get confused which one was B and which one was T. Okay, S is always in the middle only, you have kept in the middle, S is middle, but you might get confused. Just to avoid it, just write with a marker or something in the rack only, like B, S and T. Okay, and then you put 3 test tubes. In all the test tube, take 1 ml of the enzyme reagent. What is the enzyme reagent? GOD, POD, that is the glucose oxidase peroxidase reagent. Okay, in the 3 test tubes you take, in the B test tube add 10 microliters of the distilled water, in the S test tube add 10 microliters of the standard solution, and in the T test tube you add the 10 microliter of the test sample, which is unknown. Then you mix it well, and then at 37 degrees centigrade over for 10 minutes you incubate it. After the incubation, take the reading at 520 nanometer, that is the your lambda max. Okay, once you measure the absorbance OD, you write it down just like that. ODB is this much, ODS is this much, and ODT is this much. In Beer-Lambert's law, you have done in the graph plotting to verify the law, that the law is verified, right? Over here you don't have to do the graphings, you have to just measure the values and write it down in a paper. Once you get the values, then your work is very easy. Just put down in this formation and then calculate. Okay, fine. Now, what is the principle? What happens? First of all, glucose is getting oxidized by the glucose oxidase. Okay, and after the oxidation, what is happening? Glucose is converting into gluconic acid and hydrogen peroxide is formed. Okay, H2O2 means hydrogen peroxide. Okay, now hydrogen peroxide is getting oxidized by the peroxidase. Okay, that is the composition of the reagent. In the enzyme reagent, there was GOD, that is the glucose oxidase. There was peroxidase. The first glucose oxidase oxidizes the glucose into gluconic acid and hydrogen peroxide. Then the hydrogen peroxide is getting oxidized by the peroxidase and forming the H2O, that is the water, and O, that means it's a nascent oxygen. Okay, this nascent oxygen is reacting with the 4-amino antipyrine and the phenol in the buffer, phosphate buffer, phenol in the phosphate buffer, reacting with this and forming the pink color chromogen that I mentioned. If there is glucose, then only this pink color will come, otherwise the pink color will not come. Okay, so this is the reaction that is happening over there. Now this is the chart I was mentioning. So if you make a chart like this in your copy, this becomes much more easier and it becomes easy to see and there is no confusion at all. First of all, you take 3 test tubes, blank, standard and T, mark them up, then enzyme reagent, how much you are adding? 1 ml to each of them. Okay, then in the blanks test tube, what will you add? Distilled water, so that is 10 microliters, and rest of it there is no distilled water. In the standard, you will add the standard glucose 10 microliter and the rest of the 2 empty. And in the test tube, you will add 10 microliters of the test sample and the rest of the 2 is blank. Okay, whatever was written in there in the first slide, the same thing is just formatted in the chart. In some books you can get in the ml format as well. So 10 microliter is how much ml? 0.01 ml. Okay, don't confuse it. Okay, if in some books, in the bookish term, they might write it like 0.01 ml. So just so you people have a better idea, I just converted it into the microliter and written it as a microliter only. Whether it is 10 microliter or it is 0.01 ml, it's the same thing. Now calculation, the same formula that I have written over there. First you get the values, the ODB, absorbance in the blank, absorbance in the standard, and absorbance in the test. So test minus blank divided by standard minus blank into concentration of the standard. Concentration of the standard we already know, 100. So just follow the equation and you will get your result. And that's all. This is that simple test. But yeah, when you are performing it, just be cautious that you are taking exact 1 ml of sample. Do not take bubbles into it. When you are using the 10 microliters of it, put all of the samples into the solution, otherwise you will get a faulty result. Okay, so that theoretically it is easy, but practically little tricky. Okay, what we will do, anybody use one copy or just try to remember what we will do. 3 test tube, mark it with B, S and T. Let's say this is B, S and T. We have taken 3 test tubes. Okay, this is blank, standard and test. Okay, what we will do, first we will add 1 ml of enzyme reagent. Which one is the enzyme reagent? This is the enzyme reagent. Okay, so see, first of all you see how much is marked over here. 1000 microliter means 1 ml. Fine, this is for 1 ml and this for the microliters, like 10 microliters, 20 microliters. For those we will use these small pipette tips. Okay, that's this, fix this. Then after that we will take 1 ml of it. you have to just press it up to this much. After that you will get resistance, but after that also you can press, but don't press it. Okay, press it up to this much, put it deep in, slowly release it. You are getting exact 1 ml. Take the test tube, put it inside, release the sample. At the end, forcefully press it. Everything came down, take it out. Your blank filled up, standard, same way. Take the sample, put it inside the test tube. Done? Take the standard. Take the sample. Done. 3 samples. Fine. Now this enzyme reagents part is over. Okay. Now I have to add 3 samples. Okay. In the blank I will be adding distilled water. That is around 10 microliters. So how much I have fixed it over here? 10. Right? Take the pipette. Okay. This much. I'll put in the distilled water. Take the sample. See? Deep inside the test tube. Release it. So that is why if you are doing wrong process, your values will also get value. You will get the wrong values because the amount is very less. Okay, so our blank is done. I'll mix it and I'll keep it over here. Okay, now basically what you have to do, you have to use another tip for the standard, another tip for the test. Okay, but if you're taking only one tip, you are using only one tip in the exam. Let's say you are not provided by 3 tips, you have given only one tip. First always take the blank so that the mixture does not happen. Okay, after that what we will take? We will take the standard. This is the standard. Done? Done? Mix it, keep it for the test. Okay, now there are 2 types of test sample is given. One is, this is our lab made samples. Okay, and this is the real blood sample. Okay, if you're taking from the real blood sample, sample done. Okay. Mixed up, right? Now I will do what? I will take a fresh test. Okay, now I have to incubate it for 10 minutes at 37 degrees centigrade. Okay, I have to wait for 10 minutes over here. Okay, by that time you prepare one more fresh sample for you all. Okay.